Hi, my name is Mark Costa and I run the Bioactive Molecule Synthesis group at the Griffith Institute for Drug Discovery at Griffith University in Brisbane, Australia. And this is a talk that I presented to colleagues within GRID a couple of years ago now, but I think it sort of summarises some of the work that we do in my research group. So I have recorded it for anyone who's interested. So the aims of this seminar or this video are to convince you that making molecules is important and that advancements in organic synthesis methodology are important for being able to make molecules in a more efficient manner. And that secondly, we can influence biology through modern synthetic organic chemistry and that can facilitate biological investigations. So bioactive molecule synthesis at GRID within my group canvasses two main areas. There's natural products chemistry with a particular emphasis on medicinal chemistry around these natural products. We were the first group to make an anti-cancer compound named Angelmarin. We worked on medicinal chemistry around the anti-malarial compound thioplocortone, and we've recently been doing some chemical biology investigations around the natural product selenomycin, which is cytotoxic towards cancer stem cells. But we also are involved in synthetic methodology development, and this could be possibly lead to focused libraries as part of drug discovery programs. And we're particularly interested in three-dimensional scaffolds for drug discovery. So the first part of my talk, making molecules. So the drug discovery pipeline really has 6 stages. Firstly, you identify a target. Once you've identified that biological target, you can then discover a lead through maybe high-throughput screening, or these days maybe fragment-based drug discovery. We then try to optimize the lead. We try to make it more potent, make it less toxic, more soluble, and so on. We then progress that to a candidate drug that's taken into animals, and then we take that through to investigational new drug status, where we can then proceed to clinical trials, hopefully, with a very small number of compounds out of the whole pipeline. And then if you're very lucky, you get to lodge that new compound for use in humans as a new chemical drug entity or a new drug application. So what we really need in this process, I believe, are better leads for that lead discovery part of the process, number 2 there. So better leads are important because compound libraries are used in high throughput screening as a traditional starting point for drug discovery. And at the moment, I believe they're heavily skewed towards what can be made easily. So they're heavily skewed towards flat ring systems, they have no stereogenic centers, they're largely two-dimensional. However, there's been interest in recent years in going towards more three-dimensional molecules. So I'll just make this a little bit bigger. So there's been some papers published by Frank Lovering from Wyeth, and he's entitled these papers Escape from Flatland. So Escape from Flatland is really alluding to the fact that we're trying to get away from flat molecules and towards three-dimensional molecules. There's been 2 major papers in this area. I've just put the title pages and abstracts up here for your benefit. So these papers really got us interested in this area of drug discovery and how to make better drug libraries for screening. So really diversity in 3 dimensions is preferable. If you look at the number of isomers of pyridine, there's just 5 isomers of pyridine where we put 2 methyl groups on the ring. Molecular weight is 107, and one way of looking at this 3-dimensionality or this flatness is through fraction of sp3 hybridized atoms. So a fraction of sp3 in dimethylpyridines is 0.29. Now if we change that and look at the saturated system, dimethylpyridines, there's 34 isomers. They have a molecular weight not much more, but they have a fraction of sp3 of 1 because we have no points of unsaturation. And so each of these molecules are highly three-dimensional. I would argue that they cover a much greater area of three-dimensional chemical space. Now, why is it preferable to have a three-dimensionality to your compounds? Well, it was shown by Lovering that as you go through the development pipeline, the fraction of sp3 centers increases as we go further and further along the pipeline. So at the discovery stage, the fraction of sp3 averages around 0.35, but by the time we get to actually approved drugs, we're looking at 0.47. So it's quite a substantial increase in the amount of saturation within these molecules, and probably correlates quite well with three-dimensionality. We can look at this in more detail in terms of solubility. So as you increase the fraction of sp3 within a molecule, on average the solubility increases. So very insoluble compounds tend to be those that are quite flat, and we know why this is the case. They tend to stack well, form crystals that are very stable, very slow to dissolve, or very recalcitrant to dissolve from a thermodynamic point of view. Lovering also looked at the promiscuity of compounds against other off-target enzymes versus the fraction sp3. And he showed the promiscuity goes down as we move to more three-dimensional or higher fraction sp3 compounds. So the promiscuity in the highest band, around 0.9 fraction sp3, the promiscuity is actually very low. You can also look at this in terms of chiral centers, which is another big interest of mine, is chirality. This is, I guess, inherently an aspect of three-dimensionality. As we go to larger numbers of chiral centers, we also get to lower degrees of promiscuity. So this really reminds me of a joke that I quite like. So this is the gentleman who's lost his wallet. He may be drunk in some versions of the joke. And the policeman stumbles upon him looking for his wallet, and the policeman says, well, is this where you lost your wallet? And he says, no, I lost it in the park, but the light's much better over here. So I think this is what we're doing with drug discovery around the kind of two-dimensional libraries that are currently available. We're making these two-dimensional libraries because they're easy to make, but they're not necessarily the best libraries for drug discovery, for high-throughput screening. So three-dimensional compounds that I'm interested in really primarily are spirocycles. So these are compounds that have two rings joined together through a central atom. So they look like this. They have those two rings joined through a central atom, and I find them fascinating structures. So the most common type of spirocycles are spiroacetals, and these are really common within natural products. They're common in natural products from simple compounds from insects that are used to signal amongst themselves as pheromones, to very complex molecules from marine sources such as spongistatin-1, which has two spiroacetals within the structure, and those spiroacetals have been shown to be absolutely necessary for the biological activity of this molecule. They're present within other marine natural products such as azospiracid. But there's other types of spirocycles, such as this spirohemiaminal ether, which is also present within the azospiracid structure. There's other spirocyclic structures, such as spirocyclic ethers, present within polymaxenolide, and spirocyclic amines, such as present within panaic acid. So traditionally, spiroacetals have been made through formation of CO bonds in the ring-forming steps. So we would take a monocyclic or acyclic compound that has ketone or latent ketone functionalities and pendant hydroxyl groups, and then do acid-catalyzed cyclization to form the ketal or acetal structures. Our approach is to reverse this and to look for CC bonding events in the ring-forming step, through using high-energy intermediates, which are carbenes or metallocarbenes, and doing insertions into CH bonds that are activated by those neighbouring oxygen atoms. So when you embark on a new project like we did when we went on ahead with this a few years ago, you try to think of what could go wrong, and we really thought that a lot of things could go wrong with this particular approach. We could get dimerization of the carbenes. That's a really common sort of side reaction. We could get cyclization of the carbene onto the oxygen heteroatoms through the lone pair to form oxonium ylides, which are very common sort of chemistry that's been exploited by a number of chemists around the world. We could get OH insertions if we have hydroxyl groups present in the molecule or adventitious oxygen. And depending on the structure of the carbene, we might get beta hydride elimination to get us alkenes. So lots of things could go wrong. So initially we decided to look at substrates that would hopefully limit this, these side products. So our substrate synthesis started with this route here. We took a Grignard reagent. We then reacted that with benzaldehyde over here to give this hemiacetal. We react that with potassium hexamethyl disilazide, and the lactol potassium alkoxide reacts with this alpha bromo amide to give a very highly selective reaction to give mostly the cis stereochemistry of this particular hemiacetal. Sorry, of this particular acetal. So we get to the cis stereochemistry of that particular compound. And then we react that with a Grignard reagent to give, through displacement of that winder of amide, we get the desired precursor to the diazo compound that will then turn into the metallocarbene for CH insertion. We also looked at a homologated version of this where we have a reaction with this species here to give us a beta keto phosphonate, and then a Horner-Wallis-Lemons type reaction to give a alpha beta unsaturated ketone that's ready for also diazo transfer and then formation of metallocarbene. Okay, so we treat that compound with para-acetamido benzene sulfonyl azide, which is a really nice reagent for doing diazo transfer to activated compounds. We have nice yield of the diazo compound ready for this key CH insertion step. So when we tried that CH insertion step with a range of different rhodium catalysts, unfortunately we never were able to isolate any of the desired spiroacetal structure. So what did we isolate out of this reaction? Well, after much head scratching and a lot of NMR, we found out that we'd made this spirocyclic orthoester structure. So this particular molecule was a bit of a surprise to us at first, but part of our structural elucidation was helped by having seen similar sort of chemistry being observed by other groups doing CH insertions adjacent to activating oxygen groups. We isolated these spirocyclic orthoesters as a mixture of diastereomers where the ratio depended on exactly what catalyst we used for this particular reaction. The best results we got, and the yields aren't spectacular, the best results we got were with the most electrophilic catalyst, which is dirhodium tetratrifluoroacetate. So these two isomers differ at the anomeric carbon in their relative stereochemistry. And we needed to grow crystals of one of these and determine the stereochemistry through X-ray crystallography. So the X-ray crystallography allowed us to assign the stereochemistry of the major isomer. The minor isomer we assigned on the basis of its NMR spectrum and by a process of elimination. Okay, so how does this side product form? Well, we entered into a collaboration with Elizabeth Krensky at the University of Queensland to do DFT calculations and look at the mechanism of this reaction in detail. So in the literature we'd seen some similar chemistry being speculated as being formed, the products being formed through hydride transfer, and that hydride transfer was formulated in the literature initially as being hydride transfer to the rhodium metal. So in our collaboration with Elizabeth, the first thing that we were able to rule out was this putative hydride transfer to the rhodium metal. Much lower energy transfer was to the carbene-carbon, giving us a rhodium-bound enolate structure. That rhodium-bound enolate is then able to reorganize and then undergo CO bond formation to form the final spirocyclic orthoesters. And depending on which face of the oxonium ion that is formed when we transfer the hydride So here we've got the hydride transfer transition state. Here we've got the oxonium ion that's formed through that hydride transfer and the rhodium-bound enolate. Now that oxygen can recombine onto either face of that oxonium ion to form the two isomers that I showed you earlier. And the geometry of the double bond within those molecules is determined by the hydride transfer at this point of the mechanism here. Okay, so having found these problems with isolation of those spirocyclic orthoesters, we reasoned that if we change the structure of the diazo compound, we may get around this problem. And at the same time, we also want to have a simpler system, so pare it back to its really basic, simplest functionality. Now, if we use a diazoester and we get hydride transfer, then the enolate that's formed can recombine on carbon to form a 5-membered ring, or it can recombine on oxygen to form a 7-membered ring. And we really reasoned that the 5-membered ring would be preferable in this case. And so no matter what the mechanism, whether it's a concerted CH insertion mechanism, or whether it's a hydride transfer followed by a recombination of the oxonium ion with the formed enolate, in either case we should get the spiroacetal through CC bond formation. And indeed, when we managed to make the substrate for this chemistry, we isolated two compounds in quite good yields, and in a ratio of about 3:7 or 50-50, depending on which particular catalyst we used. So with rhodium acetate or rhodium octanoate, we get 30-70 mixture of A and B diastereomers, and we proved the stereochemistry of these once again through X-ray crystallography, this time by reduction of the ester and then derivatization with benzoyl chloride to make the paranitrobenzoate. If we use a chiral catalyst for these spiroacetal forming reactions, now the substrate is racemic, so we may speculate about how this would result in enantioselectivity, but we do see small amounts of enantiodiscrimination or enantioselectivity in these reactions with the main catalyst developed by Hugh Davies at Emory University. If we take it down to low temperatures, the best we can get is a 60% EE for the B diastereomer in these reactions. And I'd like to acknowledge Professor Jonathan White at the University of Melbourne for doing the crystallography on this part of our project. So back to our aim of making molecules that are three-dimensional for drug discovery for high throughput screening or for fragment-based drug discovery, we then took this simple three-dimensional spiroacetal and made a small library of spiroacetal structures by derivatization. So if we take that compound and reduce it with lithium aluminium hydride and then couple it under EDC conditions with a carboxylic acid, we made a small library of ester compounds based on that spiroacetal scaffold. We also reduced the ester down to the alcohol and reacted with a bunch of isocyanates and base to give us a carbamide library. And then we also hydrolyzed the ester with lithium hydroxide and water and coupled it under EDC conditions with amines to give a library of amides for drug discovery. Okay, so that's interesting, but can we go one step further and instead of starting with a molecule with one ring and then building the second ring to form the spiroacetal, can we do the reaction in a bidirectional sense? So we take an acyclic precursor and we do two CH insertions, the first to form the first ring and the second to form that spiroacetal structure. So overall, it would be a double CH insertion bidirectional approach from acyclic compounds through to complex three-dimensional molecules that are ripe for drug discovery. So we started by looking at diazoesters again, and we set these up with the CH groups undergoing insertion being just the right distance from the diazo precursor such that it will form 5-member rings, which are very favoured by CH insertion reactions under rhodium catalysis. And under the simple conditions of diradium tetraoctanoate, indeed we're able to get the desired compounds. So under those conditions, we were able to isolate 3 spiroacetals. 1A labelled here is C2 symmetric, 1B is C1 or non-symmetric, and 1C is another C2 symmetric compound. And the A and C compounds just differ in the relative configuration of this ester group relative to the oxygens within the ring. So this one has that ester group trans with respect to the oxygen within its same ring, whereas this one has that ester group cis with respect to that oxygen within the same ring. So under diradium tetraoctanoate conditions, we get a 4:1:1 ratio of those 3 compounds. We get a little bit of some side products. It's always very difficult to keep water out of these reaction mixtures, and so we get some compound resulting from insertion into water, which is compound 2 there. And under catalysis with the tetrakis triphenyl acetate ligand, we get some of a very small amount of this compound 3, which has this interesting bicyclo structure. We also, in that case, get a different major isomer. So in the case of using the very bulky tetrakis triphenyl acetate ligands on rhodium, we get 1B as the major isomer. We're also able to extend this chemistry to using a chiral catalyst. So using the rhodium PTAD catalyst, we get an 8:4:1 ratio of the spiroacetals, and the major spiroacetal is actually formed in a 89 to 94% EE for one enantiomer over the other. So that's really interesting. You go from an acyclic molecule through to a 3-dimensional molecule with 3 stereogenic centers using a chiral catalyst in high EE. Okay, so having seen that bicyclo compound 3 in that slide there, we thought, well, can we extend this chemistry to get to bicyclo structures deliberately? And so work ongoing in this area is to try to design substrates that will make different topologies, such as bicyclo systems such as that's shown in this slide here. We've also tried to extend this chemistry initially just in the monocyclization sense into cyclizing amine or protected amine containing compounds to give pyrrolidines. And so here's one example where we can insert into a CH bond adjacent to a tosyl protected amine to give us these pyrrolidines with stereochemistry and with substituents on interesting parts of the ring. The second part of my talk is about influencing biology. So one of the areas that we've been involved in is a type of anti-cancer chemotherapy or an approach to anti-cancer chemotherapy called anti-austerity. So austerity is the tolerance towards nutrient deprivation. It's a metabolic adaptation. It could be viewed as being complementary to angiogenesis, and you could view it as a new target for cancer therapy. So a number of natural products have been shown to eliminate this tolerance. The anti-austerity chemotherapeutic approach is to try to target this pathway via these natural products or derivatives and eliminate this tolerance mechanism. Mechanistic understanding and biological targets associated with this anti-austerity approach are largely absent. We know that AKT is overexpressed in most cells that are tolerant towards nutrient starvation. We also know that antisense RNA for AKT1 and AKT2, but not AKT3, diminishes tolerance. And we've reviewed this area in 2010, particularly focusing on natural products that have some influence on this austerity pathway. So our work is focused largely on Angelmarin. This is the natural product that's been found to have anti-austerity activity, and we developed the first total synthesis of this natural product, and we developed it in such a way that is very amenable to the production of analogues for medicinal chemistry. So the final step of the synthesis is to combine a molecule with an aryl aldehyde, a benzaldehyde, through a Novanagel condensation, and we use that chemistry to make a number, over 20 analogues of the natural product, And we find some really interesting structure-activity relationships. So the natural product has a hydroxyl group in the 4 position of the aromatic ring. We find if we replace it with a methyl group, we still retain activity, although it does go down a little bit. If we cat the hydroxyl group as the methyl ether, the activity goes down drastically. However, if we cap it as the acetate, acetate ester, the activity is actually enhanced, and we speculate that may be because the molecule is now more permeable across the cell membrane. We also found that replacing the hydroxyl group with a trifluoromethyl group enhances the activity. If it's at the meta position, we get similar activity to the natural product. However, if we replace the para-hydroxyl group with a para-cyano group, we completely obliterate the activity, which we're really surprised about. And we published this medicinal chemistry work in ChemMed Chem in 2012. So here we've just highlighted some of the results of our medicinal chemistry work. We've developed the first synthetic route to this compound in analogues. It's concise and analog friendly. Some of the analogues are more active than the natural product, and we are working on the biological targets and mechanism of action along with collaborators. Another area we're really interested in is particularly within GRID, we've got a lot of chemists involved in natural product discovery. So we've got a lot of natural products with really interesting biological activity. And there's been some amazing chemistry published in the literature in recent years where you can do very selective chemistry to derivatize complex molecules like natural products and select particular functional groups through very selective chemistry. So particularly Daniel Romo in the US has pioneered this area and has reported in Natural Product Reports a review of some methods in this area. So for example, if you have an electron deficient double bond, you can use sulfonium ylides to make cyclopropanes, and you can design those reagents so they have a pendant alkyne that is then available for doing click chemistry for doing really nice chemical biology, doing target pull down, making fluorescent probes and so on. You can do rhodium catalyzed insertion or cyclopropanations to make either hydroxyl inserted compounds if you have a hydroxyl group within the molecule, or you can do cyclopropanations if you have an electron-rich alkene within your molecule. And more recently, it's been shown that if you have a tertiary amine within your molecule, you can do these rhodium catalyzed type processes with the right substrates to get to molecules that have very interesting structures, such as that are shown on the bottom left-hand side of the slide over here. So we're interested in taking that type of chemistry and applying it to bioactive small molecules and derivatize them in such a way that they are turned into fluorophores, they turn into biotin tag molecules, photoaffinity tag molecules, and then with biology collaborators, looking at how these molecules work, what parts of the cell or parasite they go to, what biological targets they're interacting with to incite their mechanism of action. Okay, so it just remains for me to thank the people who did this work and other people in my group over the last few years. So I'd like to thank Melissa Barnier, Sam James, Romain Lepage, Fanny Lombard and Pacormat Sambatak, who all worked on either the projects I showed you or other projects within the group over the last few years. I'd like to thank Peter Healy and Jonathan White for X-ray crystallography, the ARC and HMRC and Griffith University for funding, and Professor Derek Tan from the Memorial Sloan Kettering, who welcomed me into his group for a sabbatical stay in which I was able to really start off some of the CH insertion chemistry, particularly the bidirectional approach that I talked about earlier. Okay, so thanks once again. If you want to know anything more about the projects that I've talked about, feel free to drop me an email or go to my website and find out more. Thanks a lot.